The first secondary lymphoid organ that we will be talking about is lymph node. Lymph nodes, or glands, are the bean-shaped organ that constitute the main bulk of the organized lymphoid tissue. They are 1 to 15 millimeter in diameter. They are strategically placed so that lymph from most parts of the body drains through a series of nodes before reaching the thoracic duct. Now, there is a right lymphatic duct that drains the right upper quadrant of the body, while the thoracic duct drains the remaining part of the body. Now, this thoracic duct then empties into the subclavian vein to allow the lymphocytes to recirculate again via the blood. Here, if you have a closer look at the structure of lymph node, you will see incoming lymphatics, which bring lymph into the node. These are known as afferent lymphatics, or AL for short, while the outgoing lymphatics, through which lymph exits the node, are known as efferent lymphatics, or L for short. In addition, there are small arteries, or arterioles, carrying the blood that nourishes the cells of the lymph node, as well as veins, through which this blood leaves the node. If you look carefully at this figure, you can also see the high endothelial venules. Keeping this picture in your mind, can you see how lymphocytes, both B and T cells, enter a lymph node? That's right. They can enter from the blood by pushing their way between the cells of the high endothelial venules. There is also another way. Lymphocytes can enter the lymph node, and that's with the lymph. Although lymphocytes have two ways to gain entry to a lymph node, but they only exit via the lymph, because those high endothelial venules won't let them back into the blood. Since lymph nodes are places where lymphocytes find their cognate antigen, we also need to discuss how this antigen gets there. When dendritic cells, stationed out in the tissues, are stimulated by battle signals, they leave the tissues via the lymph and carry the antigen they have acquired at the battle scene into the secondary lymphoid organs. So, this is one way antigen can enter a lymph node. In addition, antigen which has been opsonized, either by complement or by antibodies, can be carried by the lymph into the node. There, the opsonized antigen will be captured by follicular dendritic cells for display to B cells. When lymph enters a node, it percolates through holes in the marginal sinus, through the cortex and paracortex, and finally into the medullary sinus, from where it exits the node via the outgoing lymphatic vessels, or we call them efferent lymphatics. Now, the walls of the marginal sinus are carpeted, which reduces the number of invaders that the adaptive immune system will need to deal with, and it helps keep these pathogens from entering the bloodstream. This is important because blood can carry invaders throughout the body, potentially turning a localized infection into a systemic one. So, an important function of a lymph node is as a lymph filter. We have already discussed that the high endothelial venules are located in the paracortex. So B and T cells pass through this region of the node when they arrive from the blood. T cells tend to accumulate in the paracortex, being retained there by adhesion molecules. So we name this area as T cell area. This accumulation of T cells makes good sense, because dendritic cells also are found in the paracortex. And of course, one object of this game is to get T cells together with these antigen-presenting cells. On the other hand, B cells entering a lymph node accumulate in the cortex, the area where lymphoid follicles are located. So you can refer this area as B cell area. This localization of B cells works well, because the follicular dendritic cells that display opsonized antigen to B cells are located in this region of the lymph node. So, a lymph node is a highly organized place with specific areas for antigen-presenting cells, T lymphocytes, B lymphocytes, and macrophages.